Hi, yeah, my name is Jesse, and I'm going to be talking about estimating the effects of mutations to SARS-CoV-2 proteins just using natural sequence data. And this is collaborative work with Richard Nayer, who's pictured here. And I posted the link to these slides in the chat. So as I think everyone appreciates, determining the effects of viral mutations is important. And during the COVID-19 pandemic, it's been important for at least three reasons. One is that new variants arise with mutations in the genome, and it's very valuable to be able to interpret the consequences of those mutations seen during SARS-CoV-2 surveillance. For the purposes of sort of these AViD grants, such information can be important for informing the design of drugs, because we now know that SARS-CoV-2 can rapidly evolve resistance to at least antibody therapeutic drugs. And so designing those drugs to target constrained regions of the virus can reduce the chance of resistance emerging. And then finally, understanding the effects of mutations is just an important and classic way to understand the function and mechanisms of viral proteins. So what is the traditional way to determine the effect of mutations? So the traditional way to do this is experiments. So basically you have some viral gene of interest. You will take this viral gene and you will engineer some mutations into that viral gene. You then might generate either viruses or viral proteins, depending on exactly what you're studying, that contain those mutations. And then you do an experimental assay in the lab to measure the phenotype of that mutated virus or viral protein. So in this cartoon example, maybe the unmutated viral protein functions quite well. The orange mutant still functions well, but the green mutant is somehow deleterious. And so we would say that there's strong constraint or selection against that green mutation. And most of the work in my research group has centered around trying to do these experiments at very large scale via a technique called deep mutational scanning, where we essentially try to make measurements of the effects of mutations to viral proteins in very high throughput using techniques like yeast display or viral libraries. And so I certainly still think this experimental approach is very valuable. But I'd like to point out there are some limitations of using experiments to understand the effects of mutations to viral proteins. First of all, experiments are very laborious. So over the last 3 years, a lot of people have been studying mutations to SARS-CoV-2 proteins, but we still only have large-scale measurements for two proteins, spike in its receptor binding domain and MPRO. And then we have a small number of experimental measurements to mutations to some of the other viral proteins. But there's still many, many SARS-CoV-2 mutations that haven't been characterized at all experimentally, despite this intense study. Second, laboratory assays measure the effects of mutations in some laboratory system, such as cells or mice or something like that. They don't measure the effect of the mutations in humans, which in some cases is what we would be most interested in knowing. And finally, there are some viral proteins that have poorly understood functions, such that we do not even really have good laboratory assays for what these proteins do. However, if we think about this, natural evolution has sort of been conducting exactly the experiment that in many cases we would like to do. So nature is essentially testing the effects of viral mutations in humans all the time. So when a person is infected with SARS-CoV-2, many viral mutants are generated within the body of that infected person. And most of those mutants sort of never have a chance to propagate onward, but the typical person will infect, one or two or sometimes more other people. And when that happens, essentially what happens is they're exhaling some respiratory droplet with the virus and it's, landing in the airways of another person. And the chance that a virus is successfully transmitted in this way will depend on the mutations that it contains. If it contains deleterious mutations, it's not going to transmit. If it contains beneficial mutations, it may transmit better. So in a way, nature is making these measurements of the effects of mutations all the time. And in fact, if you do the math, we can see that the average neutral single nucleotide mutation, so this is a mutation that does not have a fitness effect on the virus, has occurred about 15,000 independent times in human transmitted SARS-CoV-2 over the last 3 years. And so that's a huge number of times. And so nature has not only done this experiment that we'd like to know the result of, nature's done the experiment many, many times. And just to tell you how we get to this number of 15,000, the viral substitution rate at synonymous sites, so sites where mutations don't affect the protein sequence and typically don't have large fitness effects, is about 7.5 times 10 to the minus 4th per year. The typical SARS CoV-2 infection lasts about 0.01 years. And as of early 2023, there's probably been on the order of 6 billion infections around the world. So you multiply those all together and you get that in human transmitted SARS CoV-2, in the absence of selection, the average neutral single nucleotide mutation site will have been mutated about 45,000 times. Each site can then mutate to 3 other things. So any given mutation has occurred at an average of about 15,000 times. I'd also like to note that the mutation spectrum of SARS CoV-2 is highly uneven. So some mutations have sort of been tested by natural evolution much more than others. So for instance, a typical C to T mutation has occurred about 50,000 times in human transmitted SARS CoV-2, whereas A to C mutations have occurred only about 1,000 times. And so sort of the key thing that underlies the motivation of the approach I'm talking about is that there are now enough publicly available human SARS-CoV-2 sequences that we can start to read out the effects of viral mutations on human transmission just by looking at these natural experiments. So in the work I'm going to talk about, we're using the 6.5 million publicly available sequences in the Usher mutation annotated tree. And these sequences represent about 0.1% of all human SARS-CoV-2 infections as of early 2023. So most of the virus's evolution still has not been observed, but enough of it has now been observed that we can start to draw useful conclusions. And so in particular, the essence of our approach is first to calculate how often each nucleotide mutation is expected to be observed in the absence of protein level selection. And so to do that, we look at the fourfold synonymous sites in the viral genome. So some of the codons have third codon positions where no matter what nucleotide you put in there, you get the same amino acid. So the evolution of these sites is not affected by protein level selection. And in general, the changes you see at these sites is pretty reflective of the underlying viral mutation rate. So what we first do is we look across all of these 6.5 million sequences and we count up how many times each mutation has occurred at these fourfold degenerate sites. And really importantly, we're counting the unique occurrences of mutations, not the number of sequences with the mutation. So during evolution, you can often get a mutation that occurs once, as indicated here, that's then observed in several different sequences just due to common descent because that sequence expanded. And what we really want to ask is how many times did that mutation occur and spread at least sort of once to be sequenced versus what's expected. So we're counting these lightning bolts. We're counting the occurrences of mutations, not the mutation frequency in the final alignment. So here we would count 3. And so if we do that counting at the fourfold degenerate sites and sort of average across these sites, we find that the average C to T mutation is expected to have been observed about 500 times in human transmitted SARS-CoV-2 in the absence of selection. Some other much rarer mutation types like A to C are only expected to have been observed about 8 times. And in general, you can imagine the estimates you can make are going to be more accurate, sort of the more expected counts you have of a mutation, because there's less statistical noise. In general, I'm going to draw a threshold at 10 and only consider mutations with at least 10 expected counts. So that's how many times you expect these mutations to occur based on these fourfold degenerate sites. If we actually look at the synonymous sites in SARS-CoV-2, we can see at synonymous sites, mutations occur about as much as expected on average. However, non-synonymous mutations, which change the protein sequence, are observed to occur less frequently on average than expected, because in some cases, changing the sequence of SARS-CoV-2 proteins is deleterious to the virus. And then stop codon mutations, which are often highly deleterious, are observed to occur much, much less than expected given the underlying mutation rate, because stop codon mutations are often highly deleterious. So here I'm sort of showing this calculation averaged across all the sites. But the key thing is that there are enough sequences that we can do this calculation on a per mutation basis to estimate the effect of each mutation. So we calculate the effect of a mutation as a logarithm of the actual versus expected mutation counts. So an effect of 0 indicates that the mutation is observed in human transmitted SARS-CoV-2 as often as you'd expect it to occur. And that will have an effect of 0. And lower expected or actual than expected counts will give you a negative score, which indicates a deleterious mutation. And as I noted, the estimates are more accurate for mutations with larger expected counts. So here is now the distribution of estimated effects of all mutations to SARS-CoV-2 codons. So synonymous mutations typically have an effect of around 0, as expected, because most of these mutations are about neutral. Non-synonymous or amino acid mutations, some of them are neutral and have effect around 0, but many of them are also highly deleterious. And then the stop codon mutations are almost always strongly deleterious. Now, if you look at this, you might say, well, what about these, small number of stop codon mutations that appear to be well tolerated? Well, this plot is showing all of the viral proteins. If we look at just one like NSP12, which is the RNA-dependent RNA polymerase and is expected to be under very strong selection, we can see that stop codon mutations are always deleterious to NSP12. Amino acid mutations are usually quite deleterious because this is probably a very constrained protein. If we look at some other protein like ORF8, which is an accessory gene that sort of has a poorly understood function, as it has in fact even fixed stop codon mutations in some human lineages, we can see that there's essentially no amino acid constraint on ORF8 and very little selection even against stop codons in ORF8. And to sort of aggregate that type of analysis across all of the viral genes, here I'm plotting the median and interquartile range effect of synonymous, non-synonymous, and stop codon mutations to each of the viral proteins. So if we first just look at the synonymous mutations, we can see that for almost all of these viral proteins, synonymous mutations tend to have about neutral effects of about 0. There's a few proteins like E and ORF7b, where some of them are a little bit deleterious. And we think that could be because there's important elements of RNA structure in the genome in those regions. When we look at stop codon mutations, we can see that in all of the non-structural proteins, the NSP proteins, which are involved in sort of the viral replication complex, stop codons are highly deleterious. They're also highly deleterious in the structural proteins M, E, S, and N. But when you look at these accessory proteins, which in general have poorly understood functions and are quite variable across different coronaviruses, you see that even stop codon mutations either have no deleterious effect or only very slight deleterious effects in most of the accessory proteins. In fact, the only accessory protein where stop codon mutations are strongly deleterious in human transmitted SARS-CoV-2 is in ORF3. And this actually corresponds quite well to experiments. So for instance, Matt Freeman's group recently published experiments where they created actual SARS-CoV-2 viruses knocked out in ORF3, ORF6, ORF7, or ORF8 and tested them in human ACE2 transgenic mice. And you can see that only the ORF3 knockout clearly attenuated the virus in the human ACE2 transgenic mice, which corresponds to ORF3 being the accessory protein where there's the strongest selection against stop codon mutations in human transmitted SARS-CoV-2. And then if we look at the level of amino acid constraint, we can see that, for most proteins, there's some level of amino acid constraint, except for these accessory proteins, some of them, but some of the proteins are under more amino acid constraint than others. So obviously, if you were to design a SARS-CoV-2 drug, you definitely would not want to make it against any of these accessory proteins because there's a lot of capacity for evolution at the protein sequence there. And among the non-structural and structural proteins, in general, proteins with higher levels of amino acid level constraint, like NSP13, might be a better drug candidate than something with less amino acid level constraint, like NSP1, if your concern is to avoid resistance. But really, if you're doing drug design, you probably want to look in detail at the mutation level. So first here, I'm just showing ORF3A. So recall ORF3A was this viral protein for which there wasn't really much selection at the amino acid level, but there was selection against stop codons. And this would correspond to the experiments by Matt Freeman's lab. So for instance, if we look at the effect of stop codons, so just to orient in these plots, these heat maps are showing the estimated effect of all of the single nucleotide accessible amino acid mutations, red being bad, blue appearing to be slightly beneficial, white being about neutral. And then the line plot is showing the average effect of mutations at each site. So since ORF3A is only under very weak selection in humans, there's almost no amino acid level constraint at most sites. But you can see stop codons are still deleterious up until about the last 40 amino acids of the protein. So we would predict if you were to put a C-terminally truncated ORF3A in mice, the virus would not be attenuated. You can see there's a few regions of ORF3A, such as sites like 138, where there is pretty strong selection against mutations. So whatever ORF3A is doing, those are probably some of the key sites. But anyway, ORF3A is not a good drug target. But let's look at something like NSP6. So this is now something you could reasonably think of as a drug target, because if you look across the sites, there's quite a bit of amino acid level constraint. The average effect of mutations is deleterious at most positions in NSP6. And corresponding to that, there's very little blue and a lot of red in these heat maps. And so, for instance, if you're using some sort of rational drug design approach, I know nothing about NSP6, but you might want to identify regions like this one, where sort of all of the amino acid mutations are strongly deleterious as good targets for drug design, not regions like right here, where there's actually sort of a number of mutations which appear to potentially even be slightly beneficial to the virus, or at least fairly neutral. And this is a region that's more likely to change during natural evolution. So anyway, we hope that sort of having these maps can be an informative input at the early stages of drug design to really ensure that drugs are targeting mutationally intolerant parts of the virus. I'd also just like to briefly mention some additional aspects of these analyses. First, you can see the estimated effects of mutations are quite consistent if we look across sequence subsets from different locations. So for instance, here showing just sequences from England or just sequences from the USA and making the estimates, and they're very well correlated. I discussed how if you increase the stringency of how many counts you require, how many expected observations of a mutation you require to make an estimate, the correlation gets better, but it does so at the cost of making estimates for fewer points. So like here at a threshold of 32, the correlation coefficient is higher, but the number of mutations is lower. And you get more mutations with estimates with a worse correlation coefficient if you adjust downwards. Another question people often ask is, how much are the effects of these mutations changing during viral evolution? So here I've correlated the estimated effects of mutations in Delta versus several of the Omicron lineages. So you can see the estimated effects of mutations overall are pretty well correlated between Delta and Omicron, although there is some spread. So if we look just at the estimated effects to the non-structural proteins, so things in so-called ORF1AB, you can see that correlation is a little bit higher. There's not many points strongly off the diagonal. But if we look at the estimated effects of mutations in spike, we can see the correlation is worse. So that would suggest the effects of mutations in spike have changed a little more as the virus has evolved than in other proteins. And that makes sense for two reasons. First, spike has gotten the highest total number of mutations. So as a protein gets more and more mutations, that can shift the effect of new mutations. That's happened more for spike than other proteins. And second, the actual selection pressure on spike has probably changed over time. So for instance, comparing Delta to Omicron BA.5, we can see that a mutation like, let's say, what's this one right here, K444N appears to be quite beneficial in Omicron BA.5, but not really beneficial in Delta. And that makes sense. K444N is known to be an antibody escape mutation. Probably there wasn't so much antibody pressure when Delta was circulating because most people are naive. But now in the era that Omicron's circulating, there's a lot of pressure to escape antibodies with mutations such as that one. And then finally, I'd like to note that these estimates that we can make from natural sequences correlate quite well with experiments. So here, I'm going to just show the common set of mutations. And our lab has done 2 different deep mutational scannings of SARS CoV-2. One using a lentiviral pseudotype platform of the whole spike, and one using yeast-displayed deep mutational scanning of the receptor binding domain. And if you look at the mutations that are measured in common in those 2 experiments, are 2 different experimental methodologies. The measurements correlate with each other with a correlation coefficient of 0.73. And that's almost as good as the correlation coefficient of these fitness estimates from natural sequences with either of the experiments. So I think that this suggests that these estimates are in comparable accuracy to experiments and are probably worth, viewing as almost experimental quality measurements. And finally, we just have this link here, which is linked to in these slides and also in our preprint, where there are now these heat maps of the effects of mutations for all of the SARS-CoV-2 proteins. So for instance, if you're doing drug design targeting, one of these proteins that's say NSP6, you can click here and go to this heat map and get these estimated effects of mutations and look very quickly how much functional constraint there appears to be on that site in humans. And so we're hoping that can be a useful resource. And anyway, that's the end of my talk. I'd like to again thank my collaborator, Richard Nair, and take any questions.